Welcome everybody to the lunchtime session for the Research Symposium here at Salt Lake Community College. My name is Jonathan Barnes. I'm from the Division of Natural Sciences and Engineering, and I'm pleased to announce our guest speaker for this hour. So grateful that she was able to spend some time with us, and I know that she's been inspecting a little bit of your posters. I think the feedback was good, so that's all right. I've had the privilege of having to have chat with her, and I'm so excited to introduce you to Susan Manley, who is the Vice President of Medical Services at Myriad Genetics. She's a licensed genetic counselor and has been involved in the genetic testing industry for over 20 years, applying both her clinical and business training to various roles. She's held a variety of positions at Myriad Genetics, helping lead the dramatic growth of the organization to over $800 million in annual genetic testing revenues over a 20-year period. She has a Bachelor of Science in Biology and Certificate in Biotechnology from the University of Wisconsin-Milwaukee. She has a Master's in Human Genetics and Genetics Counseling from the University of Cincinnati, and a Master's of Business Administration, an MBA, and Management from Loyola College in Maryland. We're very lucky to have her with us, and we're grateful for her time. And at this time, I'd like to turn the time over to Susan Manley. Thank you so much.
Thank you. Thanks, Rob. Thank you very much. Can you hear me all right? Everything sound okay so far? Thanks for having me today. I'm very excited to be here, tell you a little bit more about personalized medicine and genetics and genetic testing. I've spent my career in genetics, and specifically in medical genetics, so less on the research side and more interfacing with clinical diagnostics and patients. My master's degree in genetic counseling is it's a 2-year master's degree, and genetic counselors spend a lot of time talking to people about genetic testing, what it means, what it doesn't mean, what it can mean for their health and their family. And then I got an MBA and went to work in an industry position to try to help make genetic testing available to more people in a very appropriate way. So I'm excited to be here because I know all of you are interested in STEM and science, engineering, technology, and math. And I'll share a little bit of my journey through genetics in that over the last 20 years, the field has changed dramatically. And I think most of the science fields similar to genetics will continue that transformation. And we'll need people interested in a broad array of sciences and a broad array of engineering and mathematic principles so that we can continue to solve some of these problems using those skills. So in the 4th century BC, Hippocrates, who's known as the father of medicine, already figured out that personalized medicine was very important by saying it's far more important to know what person the disease has than what disease the person has. And what he was saying is that there are many complex factors in any one individual that change how that disease expresses itself, that change how that person responds to that disease or to that treatment. And although this concept was interesting a very long time ago, it has taken centuries to really start to understand personalized medicine and how we can make meaningful impacts in healthcare. So traditionally in healthcare, an individual would receive a diagnosis of any kind of condition or disease, and then scientists would study them through clinical trials and long-term outcomes, and then develop a standard of care treatment, sort of a one-size-fits-all. And so if you think of a disease like breast cancer, for example, many women with breast cancer are involved in clinical trials, given similar treatments, and outcomes are measured. And this approach really plays to the average patient. There were many patients who didn't respond at all to therapies, and some patients were cured, right? And there was a broad range of responses, but the standard of care was tailored to the average patient. Today, the goal is to be much more personalized with treatments by adding a level of understanding about that diagnosis. And I'll talk a lot today about companion diagnostics and genetic testing, but really any particular feature about that particular patient. It could be race, it could be gender, it could be cellular, it could be molecular. And understanding that diagnosis in that person in a more meaningful way leads to much better treatment options. We'll talk specifically about cancer. There are many types of cancer treatments that are tailored to the specific genetic defect in that individual's cancer. And it's allowing us to make much better and more impactful treatment modalities for patients and much better outcomes overall. So even I mix these terms up, right? Personalized medicine, precision medicine. You may have also heard of pharmacogenetics. These are all just different ways of saying tailoring medical treatment to the individual, right? Like we started discussing. Looking at specific characteristics of each patient, specific characteristics of their disease, and personalizing or tailoring that treatment to them. Many people are moving away from personalized medicine. I think there was a little bit of a fear that people were believing that cancer treatment was being tailored to them, their specific cancer. And while we're not quite there yet, we're able to tailor treatment to a broader subtype of cancers. So I think the generalized term of precision medicine is gaining a little bit more traction now. And what do I mean by individual characteristics? So that can be cellular features of a disease, a specific expression of a disease. It can be a molecular subtype, so a specific gene in a particular person's cancer that we know has been affected. And so right now in precision medicine, there's a huge focus on genetics, a lot of genetic data and a lot of genetic understanding about underlying disease. And in oncology, specifically in healthcare, a large focus because cancer is literally a disease of the genome. It's genes that normally function in an individual that have gone wrong, and they now cannot control that cell growth. So a lot of the precision medicine that's happening today, and that is a reality today, is happening in cancer treatment and in the oncology space. So I wasn't sure what everyone's background would be, and so I wanted to just give a very brief overview of some of the terminology that I'll be using. So DNA, as you all may know, is really the blueprint for our body and for all of the cellular functions. We inherit one copy of our genes or our genetic material from our mother and another copy of our genetic material from our father. And we inherit those genes from mother and father, but then as our bodies age over time, our DNA changes and our DNA can mutate. And that's the underlying principle behind many cancers that we see today is genes that control cell division become changed over time. They become mutated over time, either naturally or through environmental factors or chemical hazards. And geneticists like me grew up in the age where when we studied genetics, we looked at rare syndromes that were caused by single genes that had mutations in them. Examples of those would be cystic fibrosis or sickle cell anemia, things that were easy enough for geneticists to find families where you could see the pattern of that disease throughout the family, and then study the genes in that particular individual to see which genes had mutations in them. But today, we know that genetics is far from a very rare, esoteric subspecialty in medicine. We know that genetics can impact common and complex diseases like cancer, obesity, Alzheimer's disease, heart disease. And in fact, many people think there's very little in medicine today that is not touched by some sort of genetic element, whether it's inherited or just genes in our own body that are somehow not functioning properly. There's a genetic element to almost every disease in healthcare. In 2003, the Human Genome Project was completed, and it was a hugely ambitious program that required a lot of collaboration, a lot of funding. It was $2.7 billion of government money that was spent to sequence the human genome, all 3 billion base pairs and over 20,000 genes. And there was a government race to sequence the genome and a private company race to sequence the genome, but ultimately both places succeeded. And in 2003, many researchers and geneticists and physicians thought, this is it, right? We have the Holy Grail. We have the whole genome. We understand it all. We understand all the A, C, Gs, and Ts and what order they should be in. And many of us sat around thinking, gosh, in 5, 10 years, we'll sit with our physician. They'll do a little pinprick of our finger, get a drop of blood, and tell us all the stuff that's going to happen to us. And it's going to be awesome. We're going to treat it. We're going to get rid of disease, right? Well, that really didn't happen. We're still very far from that. We're making progress, and that's what I want to tell you a little bit more about today. The main thing that's happening right now is that the cost of sequencing has gone down. It's even less than $1,000 for a genome. So in 2003, our government spent $2.7 billion to sequence a human genome. Today, you can get your genome sequenced for less than $1,000. That's how quickly the technology has progressed in genetic testing. It has become very easy to get DNA and to sequence it and to understand those A, C, G, and T letters. It's almost become a commodity, so that the easy part now is sequencing, but the very challenging part is understanding what it all means. Just last week, I looked at the Genetic Testing Registry, which is a government-sponsored database where you can go in at that website and search either by medical condition or by gene name, and it'll tell you if genetic testing is available for that gene or that condition, and it'll tell you what lab does that testing, how much it costs. There were over 50,000 genetic tests in that database, encompassing over 11,000 different conditions and over 16,000 genes and over 500 labs. This is a huge industry. It's a huge opportunity. Many laboratories are coming online because sequencing is easy, it's cheap, and there are regulations in place where you can set up a diagnostic laboratory. And many of these genetic tests are reimbursed by insurance. Not all of them. We'll talk about that at the end, but many of them are. And now, many people are saying, why pick different genes, right? Why not just sequence everyone's genome, right? Which is all 3 billion base pairs of the genome. Or why not sequence someone's exome? That's the part of the genome that's expressed as proteins. It's smaller and it's cheaper. And many people say it's more informative. So at some point, we're going to go away from picking which genes go with which conditions and just sequencing whole genomes to look for information. This is a big and important and impactful industry. But as I mentioned, it's one thing to be able to sequence a genome. It's entirely another thing to understand what it means, and then it's an even further stretch to be able to do things differently to change outcomes. But that's exactly where the science is headed. And this bridge from being able to sequence DNA to being able to prevent disease is being built. But it's very difficult, it's very challenging, there are a lot of moving parts, and it requires a lot of science, a lot of engineering, a lot of technology, a lot of math, and a lot of partnership to really make all of this useful in healthcare and preventing disease. Right now, there are 4 main areas of personalized or precision medicine that are being realized today. Predisposition testing, which is taking a genetic test to find out if you're at risk for a certain disease. Biomarkers or targeted therapies. If someone has a disease, say, for example, cancer, is there something unique about their cancer that might tailor a medical treatment to their disease? Pharmacogenomics, looking at genes that metabolize drugs to determine which drugs or which level of drugs are appropriate for patients. And then prognostic testing. If I have a disease, how aggressive is it? How deadly is it, and how should I treat it? So we'll talk about areas of advancement in each of these 4 precision medicine areas. So I'll start with predisposition. This is where I've spent much of my time over the last 20-some years, and the claim to fame really of Myriad Genetic Laboratories, which is here in Salt Lake City Research Park, where I've worked for the last 22 years. Myriad is credited with the discovery of BRCA1 and BRCA2, two genes that are involved with hereditary breast cancer. So BR for breast, CA for cancer. 1, because it was the first gene discovered, and 2, very cleverly, it was the second gene that was discovered. These were discovered by looking at families that had an unusually high amount of breast cancer. You could trace them throughout generations, and researchers looked through these families' DNA to find mutations in genes that cause breast cancer. It's an important discovery because 1 in 12 women in the US develop breast cancer. Much of that breast cancer is what we call sporadic. There's no family history, there's no link that can be discerned to breast cancer. It's unfortunately breast cancer that is unexplained. There's familial breast cancer, which is what it sounds like. There are relatives with breast cancer. Relatives may share the same DNA. They also share the same environment, and they share the same lifestyle. So we can see cancer, breast cancer clustering in families. And then hereditary breast and ovarian cancer, which is due to inheriting a mutation in BRCA1, BRCA2, or other genes that predispose someone at birth to an increased risk to develop these cancers. So we see them occurring earlier in life, and we see multiple cancers. Women sometimes develop more than one breast cancer throughout their lifetime, and we see a cluster in the family. And so with the discovery of BRCA1 and BRCA2, people could take a blood test to determine whether or not they were at increased risk to develop breast cancer, which might not sound like something you would want to do, but in fact, many people are empowered to change their healthcare outcomes by getting more mammograms, by changing their lifestyle, by taking preventative medications, or even by having preventative surgeries to reduce their risk or detect cancer early if it does occur. And the test that Myriad has on the market today is called Myriad MyRisk, hereditary cancer, understanding my risk for hereditary cancer. And in 1996, when this test was launched, it was just these 2 genes at the top, BRCA1 and 2. And since 1996, with all this explosion of DNA sequencing and better understanding our genomes, we now know that there are a variety of genes that predispose people to breast cancer, also to other cancers like ovarian, gastric, colon cancer, pancreatic cancer, melanoma, prostate, and endometrial cancer. So families that had just breast cancer segregating in them were the ones that were studied to find BRCA1 and 2. But the reality is that most family histories are not that straightforward. They contain other types of cancer. And so as gene sequencing became more common, researchers sequenced more genomes, they looked at more families. Our panel now contains 28 genes that any one of them with mutations in them would predispose people to a variety of these kinds of cancers. And people can know what their risk is and do medical interventions to try to reduce their risk or to detect cancer early. The other benefit of this kind of testing is if you're in a family history that has a lot of these cancers in it, and it's determined that a relative, say for example, has a BRCA1 mutation, and you're tested and you do not have that mutation, you don't have an increased risk for those kind of cancers, even though your family history is filled with them, right? So not only are we able to say some people are at higher risk for cancer, the test is also able in some circumstances to say some people have a lower risk to develop cancer. So very powerful, and this has been around for about 20 years. In 2017, after a lot of research done by Myriad, we added an additional component to our gene testing. So I mentioned to you that Myriad was credited with the discovery of BRCA1 and 2. We weren't involved in the discovery of any of these other genes, but we were able to add those genes to our genetic panel. This new element to our test called risk score is something that Myriad took publicly available data, was able to use a lot of statistics and math to come up with another way to estimate breast cancer risk. So if someone is negative for any mutations in the genes on the panel, we can combine their personal and family history and this additional genetic assessment, which I'll tell you about, to better determine what their breast cancer risk is. So I mentioned BRCA1 and 2 and some of these other genes. They're rare, and they're very strong, if you will. So families that have BRCA1 and 2 mutations have a lot of breast cancer. The risks occur, and women are very young when they get breast cancer. And these gene mutations are relatively rare, so frequency is low, but the risk that they convey is very high. As we sequenced more people and understood more about genetics, other genes came to the table. Some of these other genes that are included in the 28 gene panel are a little bit more common than the first ones discovered, and they have a little bit less risk. It took us longer to discover those genes. We had to sequence more families, understand more genetic information. This new element to genetic testing now is called SNP testing, SNP-based testing, single nucleotide polymorphisms, and I'll tell you what those are. They're very common. The human genome, each one of us has hundreds of just one A, C, G, or T that's changed at that position. They're very common, and the risk is very low. And any one of these genetic changes would mean very little to an individual, but once you start compiling dozens of them and the risk associated with dozens of them, then you can get a clinically meaningful risk assessment. So let me tell you a little bit more about SNPs and why they're important. So as I mentioned, it's a single change in DNA. Some of the other genes with mutations, they have big chunks of the gene deleted. They stop making a protein very early, and the protein doesn't work right. They're very serious changes in our genetic material that were easy to identify. These are just a single nucleotide change. So here, this person's sequence is AAGTCA, and this person's sequence is AACTCA. 96% of the population roughly would have a G here, but 4% of the population have a C here. What does that mean? Probably nothing in most cases. But when we study enough patients and we use mathematical principles, we can associate these changes with cancer risk, and I'll tell you about those studies. What's interesting about this kind of genetic assessment is that, so for example, the BRCA1 and BRCA2 genes, we understand the role that those genes play in cell division, and we understand that when those genes have defects in them, cells can grow uncontrollably, and that leads to breast cancer. We can really make sense of all of that. These SNPs, these single nucleotide polymorphisms, can be anywhere in the genome. We have no idea sometimes how they work, why they would influence risk, what they do, why they're important, but we know statistically they are associated with cancer risk. And this is a whole new field that genetics is taking on that is much less biology-based and much more mathematics and association and statistically based. So many of these single nucleotide polymorphisms that are associated with disease risk come from, you may have heard, genome-wide association studies, or PheWAS studies. And these are large studies, and this is why it's important that gene sequencing is relatively cheap, because they take very large cohorts. So for example, this study, that was the foundation of the research we used to develop our risk score, had over 33,000 women who had breast cancer and over 33,000 women who did not have breast cancer. And they scanned the genome of all of these over 60,000 women for single nucleotide polymorphisms. Some people have a G here, some people have a C here. And what they found was that there are some nucleotide changes that are statistically more often found in breast cancer cases than in unaffected cases. So for example, having a G instead of a T at this particular position in the genome was statistically associated with an increased risk in breast cancer. We didn't know why. We didn't know what gene it was in. We aren't sure what the biological impact is, but statistically, it is associated with breast cancer risk. Small, a very small risk. But once you find 20, 50, 60, or 80 of these, you can multiply their increased risk together to get an overall increased risk for breast cancer. So not only now does our Myriad MyRisk test look at 28 genes, tell you which genes we understand well might have mutations in them that would put you at risk for cancer, we can also do a calculation of about 86 of these SNPs, single nucleotide polymorphisms that are associated with breast cancer, and provide a lifetime risk for breast cancer. That's very precise. And for some women, we calculate that the average risk for breast cancer up to age 80 is somewhere around 10, 12%. We can calculate risk scores that are as high as 50% for some women because they have so many of these single nucleotide polymorphisms that are associated with breast cancer risk. So that's where the field is heading. Like I said, way back in the day, we studied families, and we studied individual families, and we hand-selected them, and we combed through their DNA. Now, people go to genome databases and harvest thousands of genomes and search through them for interesting findings, and then try to understand what the disease risk really is. It's totally turned on its head. It's gone from less of a biology, I mean, certainly you have to know that, but so much math and statistics and approaches in that way to better understand genetic science. This just was published in 2018 in March, and this really was amazing to me, and again, I think representative of where the field is going to do. We just talked about genome-wide association studies. Now they're talking about phenome-wide. So phenome, so your genotype is what's in your genes. Your phenotype is what's expressed. So a genotype would be BRCA1, and the phenotype would be breast cancer. So what this group did at Vanderbilt is they looked at their electronic medical record. And this is exactly what I think people were hoping for when we started to put all of this health information in an electronic record. They started a study, they've enrolled patients, and they've had over 225,000 patients where they've done comprehensive genome testing and genome sequencing. From this group, they took over 21,000 patients, and they looked through their electronic medical record, and they looked at the symptoms that were listed in their health file. And then they compared those to symptoms that they had characterized in over 1,200 genetic diseases. And they found just by looking through the medical record in this patient population, 807 patients who had matching DNA analysis that had mutations in genes linked to 17 diseases. When they actually went back and looked to see if those patients had genetic testing, only 8 of those patients actually had a diagnosis. The rest of them either did not have a diagnosis or their physician had pursued genetic testing for the incorrect disorder. So just by volunteers getting their genome sequenced and looking through their electronic medical record, they were able to find hundreds of patients with genetic conditions, and those patients didn't even know. Their healthcare providers didn't know. So, like I said, the whole science is being turned on its head, from studying carefully crafted family histories to looking through enormous data sets to try to find patterns and to try to find people who are at increased risk for various conditions or who have various conditions who don't even know it. So very exciting and very interesting change in our field. So now let me tell you a bit about personalized medicine and targeted therapies. So rather than talking about who's at risk and how do we find who's at risk, these are targeted therapies are people who have a disease where we can treat them with something different based on what we know about their disease. One of the most successful examples of this is a drug called Herceptin, which is used to treat breast cancer cases that have overexpression of the HER2 receptor. So this is normally a receptor that sits on the top of cells, and they stimulate the cell to grow appropriately. In certain breast cancer cases, this receptor is overexpressed, and as you can see by looking at this picture, the cell is likely to get too much signal to grow, and that's what's causing breast cancer in this individual. Herceptin was designed as a monoclonal antibody that targets this receptor and sits right on it and blocks it so that the cell no longer receives that signal to grow. And Herceptin was one of the first targeted therapies approved in breast cancer cases, and only in breast cancer cases that have this phenomenon. But it can be very successfully and well treated with Herceptin because we understand at the cellular level what's causing cancer in that particular patient. So in order to get Herceptin as a treatment, genetic testing has to be done at the tumor level to make sure it's an appropriate tumor to receive Herceptin. And so this has been a major breakthrough in breast cancer treatment, and it probably happened 8 or so years ago. It's a really wonderful success story and really opened the door to tailored treatments. There are many other examples in cancer that are similar to this. So in melanoma, there's a gene called BRAF, where 50% of those melanomas have that specific mutation, and it's an oncogene which turns on growth for that cell. So again, the cell is getting an inappropriate signal to grow. A therapy was created which is specifically targeting that mutation in that BRAF gene, which blocks the protein that it produces. So again, knowing that that melanoma has the specific gene mutation that's causing it to grow, targeting a treatment to it can successfully and effectively treat melanoma patients. And again, 50% of melanomas have that gene mutation. That's a really amazing improvement in the treatment of melanoma. Similarly, in lung cancer, there's a gene called EGFR. It occurs in 15% of non-small cell lung cancer cases. And there are therapies, Tarceva and Iressa, that are created to particularly target that type of lung cancer. A little different theory here. In colon cancer, there's a gene called KRAS. 40% of metastatic colon cancer cases have this gene mutated. And that makes it ineffective against treatment, Erbitux and Vectibix, which are commonly used treatments for colon cancer. So this is an example of where 40% of metastatic colon cancer has a gene mutation that makes treatment with the regular therapy ineffective, right? So avoiding certain treatments. These are probably the patients where in those general drug trials probably failed or didn't see good response. But the standard of care for most colon cancers remains these two therapies, but we know in some patients that won't be effective. Again, so we can avoid using those treatment modalities in those patients. And then for BRCA1 and 2, ovarian cancer is also a cancer that occurs with mutations in those genes. And there's a new class of treatment called olaparib that specifically targets the defect of BRCA1 and 2 in those ovarian cancer patients. And what's really amazing about some of these treatments, especially something here like olaparib, is that because the cancer treatment is targeted to the gene mutation in the cancer, there are not nearly as many side effects that is seen generally with chemotherapy. So as you may know, chemotherapy used to treat cancer, it's a generalized treatment that kills rapidly growing cells. These kinds of treatments target the actual cells that have gone awry, right? So not only is it more effective, it's better tolerated, there's less side effects, it's all around a much better approach to treating cancer. And while we have these approaches to some cancer, many drug companies now, when they do clinical trials, they're doing what's called basket trials. Instead of taking a bunch of people with breast cancer or colon cancer and trying different treatments, they're taking people with a BRCA mutation, regardless of what cancer they have, or PALB2 mutation, regardless of what cancer they have. Again, rather, I think the future of cancer and cancer treatment is, rather than talking about the organ in which the cancer arises, people will talk about and treat the specific gene that has gone wrong in that particular cancer cell, which is an amazing advancement. It's not a cure, but it's an amazing advancement in the way we can treat cancer. So pharmacogenomics, this is also a really important and big area of personalized medicine. There are genes involved in drug metabolism and changes in those genes where we know affects how people metabolize drugs. So if you take several patients and they each take a drug orally, they will metabolize that drug at a different rate, faster or slower, and the overall exposure that that patient has to a drug will be very different from patient to patient. One of the biggest examples here is there's a gene called CYP2C19, which is an enzyme that metabolizes drugs, and it metabolizes a number of drugs. And there's a change in that gene that means people with that gene change can metabolize certain drugs very poorly and don't respond as much. Plavix is a very commonly used drug for stroke and heart attack, to prevent strokes and heart attack. And it's on the WHO list of most essential medications worldwide, right? Very effective. But people who have a gene mutation, which is about 2 to 15% of the population, metabolize that much more slowly, and it's not as effective in those patients. And so the FDA has said, before prescribing something like Plavix, you could do a genetic test to determine whether or not someone is going to respond effectively to this therapy. Another example is 5-FU is a chemotherapy that's very inexpensive. It's been around a long time, and it's used to treat colon cancer. There's a gene that metabolizes this drug where the mutations in this gene, although they are rare, means that people can't metabolize this drug, and they get intense toxicity to chemotherapy that could be fatal, right? So again, the FDA has said before administering this drug, you should test for mutations in this gene to see if people can actually metabolize the drug correctly. So not only is it getting the right drug to the right patient, but it's avoiding certain drugs because people have differences in how they can metabolize drugs. GeneSight is a company that was acquired by Myriad Genetic Laboratories, and they have a test. The test is GeneSight. The company is Assurix Health. The test looks at a number of these genes that metabolize drugs and makes suggestions to physicians as to which drugs to prescribe for bipolar disorder, for attention deficit disorder, for depression. So right now, the approach to prescribing these drugs is sort of trial and error. A patient who has depression or bipolar disorder, the physician will start on their favorite drug to start with, right? And then the patient comes back, they're either feeling better or they're not. They try another drug, they try another drug. It takes months and months of trial and error to find the right drug for the patient. This test doesn't necessarily say this is the right drug for your patient, but it will say your patient has mutations in these particular genes, which makes metabolizing of these certain drugs challenging. And so a physician would do a lot less trial and error on these kinds of drugs for patients than what has historically been done. So again, trying to get the right drug to the right patient. And then the last of the four categories is prognostic tests. So again, these are very common in cancer, right? It can be very difficult to assess prognosis of certain cancers. And these tests are both done at Myriad. Prolaris is a test that looks at RNA in a prostate cancer. So DNA makes RNA, and RNA makes a protein. So the more RNA you have, the more protein you have. It's a different assessment of how quickly that tumor is growing. And so with prostate cancer, it's very common, and many cases of prostate cancer are not very severe. Men can live with early stage prostate cancer for a very long time with no risks or side effects. But as you might imagine, and it's not irrational, when prostate cancer and other cancers are diagnosed, people want to sort of throw the kitchen sink at it, right? Don't wait, please treat, please do surgery, right? No one wants to live with that uncertainty. But surgery and treatment for prostate cancer has a lot of undesirable side effects. And because prostate cancer is so common, there's a great desire to avoid over-treatment of prostate cancer. And what Prolaris does is look at the activity in that prostate cancer to determine how aggressive, how quickly that prostate cancer is going to grow, how likely it is that prostate cancer may metastasize, so that patients and their physicians can say, we really should treat this aggressively, or this is slow growing, it's indolent, maybe we won't treat it at all, right? So offering another level of information about the prognosis of cancer. Similarly, a test called EndoPredict actually looks at endocrine therapy or hormone therapy in breast cancer patients, whether or not that would be effective. So doing a test on a breast cancer tumor can predict the likelihood of metastasis or recurrence, which means if the score is low in a breast cancer patient, they may safely forego chemotherapy, right? And so you can imagine that's a difficult decision to make, how aggressively to treat a particular cancer. And previously it was based on size and stage of the tumor, which is still useful information, but now we can actually look at what's happening inside the tumor to figure out how quickly, how aggressive, and how deadly that tumor might be. So, as you can imagine, there's a lot of promise, there's a lot of activity, there's a lot of tests, there's a lot of companies, there's a lot of activity in this whole space. And there's a growing movement to have the FDA involved in regulating a lot of what happens in this space. So most genetic tests are not FDA regulated, but all drugs are, right? So the drug industry has to go through FDA regulation and trials to get a drug approved, but you saw all these genetic tests and all these companies, and there's a desire to sort of level the playing field, make sure the genetic test is as robust and valid as the drug is. And so the FDA has created categories called companion diagnostics, which means tests that are approved in conjunction with a treatment. So in order to get the treatment, you have to take the test, right? And so both of those elements, the treatment and the test, would go through FDA review and FDA clearance. And currently, there's just a handful of genetic tests that have achieved this companion diagnostic status with the FDA. The FDA has historically not looked at labs, not regulated labs, not looked at DNA information. It's a whole new world for the FDA. And as I showed you, there are more than 50,000 genetic tests on the market. So this is a big issue for the FDA. There's increasing pressure to regulate, but not an infrastructure to do it, and not the right knowledge base, really, from the FDA to do it effectively. They've also created another status called complementary diagnostics, which means you don't have to take the test to use the treatment, but the test will tell you who will get the most benefit from the treatment, right? So something like taking a genetic test before you receive Plavix, right? Or an antidepressant medication. Probably those tests will fall under complementary diagnostics. It's not a requirement, but it'll help you figure out who's going to get the most benefit from the treatment. So somehow, it's thought that the FDA will start looking at all of these different genetic tests and placing them in these various categories, required or recommended, really, and we'll have different types of regulatory status behind both of them. One of our tests at Myriad actually is a companion diagnostic, so the BRCA test in ovarian cancer patients for olaparib has achieved FDA clearance as a companion diagnostic, and it was a really big effort and a big deal and something we're very proud of. You may have heard recently, just a couple weeks ago, that the FDA actually gave clearance to a company called 23andMe. And they do a test that looks at 3 common BRCA1 and 2 mutations in Jewish individuals. So there are 3 mutations that occur more frequently in Jewish individuals. We call that a founder effect in genetics. It's because their ancestry is well defined and has been constricted over time, that these 3 mutations occur more frequently in people of Jewish ethnicity. If you're not Jewish, you probably don't have these 3 mutations. There are thousands and thousands of mutations in BRCA1 and 2. It was very strange for the FDA to give regulation only to 23andMe to do these 3 mutations. Plus, it was direct to consumer. Everything I've told you up until now has to be done in conjunction with a healthcare provider. This has been made direct to consumer, which I'm not opposed to. I think people should have genetic information, they should understand it, and they should use it. But it needs to be done appropriately so people understand the benefits and limitations of what they're doing. And so with that in mind, the FDA has approved this test with what they've called special controls, which basically says you shouldn't use the test results to determine any treatments. Those decisions require you to get testing confirmed and genetic counseling. And when you do all of this, you should do it with a healthcare provider. So it was a very interesting development and a sign, I think, of the pressure that FDA is under to review and regulate genetic information, to make it available to people so they can use it to improve their health, but yet all the complexities that exist here. It's very interesting, and I think this whole area is ripe for information. The FDA is eager to understand more. They're eager to have more scientists who are willing to look at this area and figure out a better way to regulate it so that it's safe for consumers. Just a quick note about reimbursement, which is important to people like me and companies like Myriad. Currently, there have been cuts across the board in healthcare spending, a lot of desire to reduce the amount of healthcare spending in the United States. And much of what I've described to you so far, even though the cost of sequencing has decreased, the ability to understand it, interpret it, and develop drugs around it remains incredibly expensive. So much of these genetic tests I talked about are very expensive. So there's a desire for third-party payers and even Medicare, Medicaid to say no to personalized medicine because it is expensive. And unfortunately, not getting reimbursed for these things is a disincentive for companies to develop personalized medicine strategies. So not only does there need to be a better and more clear, perhaps regulatory approach, third-party payers need to invest in, and this is my opinion, they need to invest in personalized medicine. It's expensive now, but over the long run will lower healthcare costs and will create a healthier population of people. And I think we'll get there. That's been the message to third-party payers, that they should invest in this. It'll pay off over time. And they're looking at changing how they reimburse physicians in general, too. As you may know, now most physicians bill insurance for services they provide. I did an x-ray. I did an exam. I did an audiology exam. I did a strep throat culture. and that's how they get paid. But some payers, like Kaiser, which is a large payer organization with a healthcare system embedded in it, pays physicians on the health of their population. Physicians will make more money if their population is healthy, versus physicians, and I don't mean to paint them as bad, just healthcare in general, making more money to do services. Right now, healthcare institutions make money on surgery. They don't make money on the fact that you don't need surgery. And there are many payers who are wanting to change that paradigm to paying for health instead of paying for disease, which fits very well into the personalized medicine future that I think we can realize. In 2015, President Obama announced a $261 million research effort called the Precision Medicine Initiative. It was a big deal. People were really, really excited about it. Initially focused on cancer, which is driving some of these developments we talked about already, but a broader goal of just improving healthcare across the population. And while in 2003 we had the Human Genome Project and we understood all the letters in someone's genome, the goal of the Precision Medicine Initiative is to understand how that all works in a person. So now, rather than sequencing a bunch of genomes, they're looking for 1 million volunteers to enroll in a database, to also provide saliva for genetic testing, but to also give information on health, disease, lifestyle, diet, nutrition, activity, right? And it's so ambitious, but it really is a desire to look at genetics and environment and lifestyle and to try to better understand a person, not just a genome or not just a disease, but how all this works in a person. So it's called the All of Us Research Program. It's in beta testing now, but it will be enrolling people spring of this year, so any time now, really. And the goal, again, is to get 1 million people enrolled and to collect safely and securely and privately, collect a lot of information on people so we can start making sense of people, not just disease. So I hope that I have imparted to you that the future for personalized medicine is really exciting. It's going to be very much driven by STEM and math and engineering and technology, but the whole of personalized medicine includes changes in our regulatory structure. People need to understand what this information means and what it doesn't mean. There need to be incentives for research and development. There's going to be a lot of intellectual property that companies like Myriad are interested in protecting while benefiting a whole lot of people. There's a whole lot of things that need to happen for us to really leverage the benefit of personalized medicine, all of which are starting now and will continue to grow over the coming years. So the whole goal of this is to try to shift the paradigm from disease and treatment to prevention, and personalized medicine can have a big role in that. Now we're able to and will continue to look for optimal therapies, avoiding trial and error for drug selection, avoiding adverse drug reactions, trying to find the specific people who are really going to benefit from certain treatments, improve quality of life, and over time really control healthcare costs and make our population healthier overall. So with that, if there are any questions about any of these areas or anything in general that you have for me, I'm happy to take them. Yes?
Is there a relationship between BRCA1 and metastasis?
I think there is some evidence that says certain mutations in BRCA1 or 2, the breast cancers that develop are more aggressive, but it's not specifically linked to metastasis. So we can't, BRCA1 and 2 aren't the genes we would look at for metastasis. The test I mentioned previously called EndoPredict, which looks at the tumor, is a better predictor of potential metastasis.
What RNA markers indicate metastasis?
Well, I'll have to get back to you on that one. I know for EndoPredict, there's 21 different RNA markers that we look at, and I don't think there's one singular one that says yes or no metastasis. It's a little bit of a complicated algorithm, but I can give you more information offline. I don't know which combination of those markers is the key one for that.
And another question on the SNPs. They're individual, or do you find covariance among them and haplotypes? And if so, are they on the same chromosome?
No, the risk score that we have has 86 different SNPs. They're all across the genome, so they're on different chromosomes. And for the SNPs, because you have 2 copies of each gene, you could have 1 or 2 copies of that SNP, and all of that is weighted in the algorithm. So the more copies of these SNPs, these 86 SNPs that you have, the higher the risk for breast cancer would be. So yes, yes, it's individual, yep. And it's all over the genome, all across many different chromosomes. Both. So there was an international research effort to discover the breast cancer genes. We certainly didn't do it all in isolation, but Mark Skolnick, who's the founder of Myriad Genetics, led the scientific team that was credited with the isolation and discovery of BRCA1 and 2. And then Myriad Genetics was formed to create the diagnostic test and make the testing available. But a lot of research occurred on BRCA1 and 2 outside of Myriad and of Mark Skolnick. But his scientific team was credited with the ultimate discovery. Yes?
You've mentioned that this can be quite cost prohibitive and that is kind of a barrier to adoption of many of these tests
Yes.
and therapies. What kind of barriers are there among the healthcare providers themselves in terms of their adoptions and their attitudes towards these?
That's an excellent question. I think that some of the newer graduated healthcare providers just coming out of medical school are much more comfortable with these principles and these concepts. Some of the older school ones are not as clear about genetics or predisposition. I think for the therapies, it's pretty widely adopted because once a therapy is approved, it goes through this FDA regulation. There's clinical trials, and they all kind of understand how that goes. Some of the predisposition tests or the pharmacogenomics, right, they kind of crop up. And I think many physicians require more education and more understanding to get to a comfort level to where they can use that appropriately. But it is a barrier. And much of what Myriad does is we do a tremendous amount of education. Just what our tests are, what they aren't, how to use them, who to use them in. It's a really big need. And in part, that's what many genetic counselors do, is try to educate and close gaps. So if physicians aren't comfortable with genetic technologies, genetic counselors can meet with patients and make certain recommendations to physicians, and then physicians can prescribe the tests. So education is a big barrier. Yes? 5FU? Mm-hmm. I know it's a funny name, isn't it? Yes, it exists. And this is one example where for the 5-fluorouracil, which is 5FU, it's a very inexpensive treatment chemotherapy for colon cancer. It's widely used, it's widely available, it's been around for a long time. Yes, yep, yes. FDA has put a black box warning on that chemotherapy to say it should not be used in people who have a DYPD gene mutation. Right on it's a contraindication. Yet the number of physicians who actually test for mutations in those genes before administering that chemotherapy is very low. They wait, and if someone has a bad reaction or side effects or a lot of toxicity, then they say, oh, maybe you've got one of those gene mutations, we'll just switch therapies, or we'll do the test then. So, that's an example of where physicians feel like I'm going to use my standard of care approach and adjust it when I see problems, where an effective and that particular genetic test is one gene, it's rather inexpensive. Doing a test like that ahead of time would avoid those toxicities in that patient. So that exists today. It's not very well utilized, though. Yes, yes. It can be ordered. Our lab does not do it, but Quest, LabCorp, ARUP, any diagnostic laboratory would do that kind of testing. Yes. Yes. I did not do the study, so it was done at Vanderbilt. I'm sure they had IRB oversight, and so probably the manner in which they did it, they could not go back and disclose to those patients who were undiagnosed, which is, I don't know that for sure. I would have to consult the study, but I'm very certain that a study like that, published in that manner, was done under IRB approval, and so either it was anonymized or patients gave consent, and they, one of those two appropriate things happened.
So in the all of me database you're trying to create, are you going to try and create it so that it is not, it's voluntary and people can get the information they need?
So that's a large government effort, one of which we're not involved with. Privacy is a very big issue in that particular study. And one of the things that the Precision Medicine Initiative stated when they announced this is that privacy and partnership with patients has to be important. People have to feel like their information will be protected, but yet people want to feel like they're a partner in that research. So I think that some of the information will be anonymized and made available generally for people to just comb and look at. Some of it, though, will be potentially disclosed back to the patient because they will legitimately find things that are very important. And so I think as people enroll in that study, part of the consent will be these things will be done with your data, which we will promise will not be traceable back to you, nor will you be able to know what happens. And then some things are important, and we will disclose them to you. Even now, studies that are done, some consent forms allow patients to say, I want to know if I have breast cancer risk. I don't want to know if I'm at risk for Alzheimer's disease. I do want to know if I have this. I don't want to know, right? Like some studies are that. I'm not sure that a study of this scale can be quite that specific, but I'm certain that they have a duty to disclose some things to these research participants, and that will be in the consent process. And it's a good thing, I think. Yes? Yes, that's a good question. Has BRCA 1 and 2 been researched in men? And yes, men can carry and do. 50% of BRCA carriers are men, 50% are women. They can get male breast cancer. It's very rare, but they're at increased risk to develop male breast cancer. They can also get pancreatic cancer and prostate cancer. So those are cancers that can appear in men if they're a carrier of a BRCA mutation. That's a good question.